Good evening. My name is Greta Samsa. I am a neuroscientist here at Columbia University. I am the granddaughter of Mr. Samsa. My object of study is the transformation, the metamorphosis itself. I believe this could happen to a human being. I am gonna donate my body to science. I will take hormones and undergo this transformation. I invite you to be part of this trip. It will take about 30 days for my whole body to transform, to become something new. Thank you for being here. I studied this for almost 20 decades. I've worked with numerous neuroscientists, endocrinologists, and other doctors. And I realized that if I mix GC with TH, the transformation can occur. I will donate my body to science. And I will go under this transformation. I will become the monstrous cockroach. And then, hopefully, I can come back and be myself.
Endocrine disruption by man-made small molecules alters hormone synthesis, metabolism, or action, and poses a great concern for human health. The use of hormone or chemical treatment of free-living humans makes them amenable to be an intact vertebrate model for the biological activity of TH receptors, agonists and antagonists. TH receptor isoform-specific actions and endocrine disrupting effects of environmental contaminants on early development in general. The in vivo effectiveness of the first TH receptor antagonist, NH3, was initially shown in tadpoles, where it blocked naturally and TH-induced metamorphic events. Likewise, the isoform-selective agonist, GC1, which preferentially binds and transactivates mammalian TH receptor beta compared to alpha, induced metamorphic changes in tadpole tissues, known in induced high levels of TH receptor beta compared with tissues that express high levels of TH receptor alpha.
This is day 7. I am undergoing transformation.
Events, as well as the timing of birth itself, are accompanied by many hormones where thyroid hormone TH and glucocorticoids GC in particular play prominent and widespread roles. Deviations from normal TH and GC signaling arising from congenital endocrine diseases in the mother or fetus, or from environmental insults, for example, nutrition stress or medical interventions, can cause deterioration, alteration in organ, maturation and timing of birth, such as in the case of Greta Samsa, as we see in Metamorphosis. I've been quite interested in investigating this case, and while taking the book Metamorphosis while reading, I decided to undergo an experiment with my own body and myself to see if I can experience some of these thyroid and glucocorticoids exchanges in my body and what it does physically and cognitively to my mind.
Molecular mechanisms of gene regulation and developmental roles of hormones are complex, involving transcriptional and epigenetic machinery that vary among cell types. Hormone interactions at multiple levels and cell-cell interactions. The study of Greta Samsa Metamorphosis has contributed greatly towards understanding this complexity and continued development of the Human Nature Toolkit, including transgenic animals and gene knockout technology, will promote future insights. Frogs, fish, and mice all have unique features with respect to humans, and some of these unique features provide valuable advantages for use in understanding the human condition. As presented in this review, the molecular, endocrine, and developmental mechanisms of TH actions and hormone interactions during metamorphosis are unique strengths of Greta Samsa's model to understand human perinatal development. Taking advantage of Greta's model is valuable because of the vital and numerous developmental roles of TH and GCs for proper development in humans, and because of the potential impact of endocrine disrupting chemicals on development.